For many of us, full and pain-free mobility is something we take for granted. Here at Loughborough University, researchers are using stem cells to bioengineer advanced neuromuscular models, with exciting applications in testing new therapies for the tens of millions of patients suffering with neuromuscular conditions worldwide. So let's find out exactly how they're doing it.
There's one thing that everybody has in common: we all have a body. But the part we're particularly interested in are bones and muscles. We want to know what happens to your muscles when you want to make them bigger, or what happens to your muscles when they get smaller, for example, if you've been injured, if you're ill.
Pretty much every single person on the planet will have a condition which compromises the function of their musculoskeletal system over the course of their life. Diagnosably, that's about 1 in 4 people. So as a consequence, there's a huge number of people that need new treatments and new therapies, and there's a real need for us to have test systems and test platforms that allow us to develop these new treatments. So if you want to develop a new treatment or a new therapy for a musculoskeletal system, then the general process is threefold. You'll start by doing some relatively simplistic tests on 2D cell culture systems. These are really effective systems for high throughput work and high throughput assays, but clearly they don't recreate the complex architecture of the human body. The second stage in that process is going into animal testing. Now, working with animals has historically provided a lot of answers and a lot of new treatments, but clearly there is an issue there around the ethics of using animals in research. And secondarily, we're talking about an animal here, we're not talking about a human being, and clearly there is a difference in the physiology of an animal versus a human being. So the research that we're doing here at Loughborough utilises bioengineering. So this is where we are trying to recreate human physiology, but within a laboratory environment. So we're recreating 3D tissues that mimic the architecture of a human being.
Andrew and Mark are working with Axel Bioscience to bioengineer neuromuscular tissues in the lab to mimic human tissue. They can even put them through a sort of gym routine using electrical or mechanical stimulation, which fast-tracks their development so they're ready for use immediately. They can then monitor changes in real time, looking for responses to potential drug candidates. The key to these advanced neuromuscular models lies in the cells, which are produced from human stem cells. These cells can be pipetted into lab platforms and matured, meaning they behave as they do in the human body and can act as an advanced in vitro platform for functionally relevant disease modelling and compound screening.
So when you're bioengineering a human tissue that recreates human physiology, the quality of the tissue can only ever be as good as the starting cells. The Axel cells that we use are high throughput, reproducible and reliable, which ultimately allows us to focus on the science and the development of new treatments and new therapies.